All right, let's step into the lab for a minute. We're going to play detective, but our crime scene, it's a chemistry bench. We've got our suspects lined up, but here's the catch. They look like identical twins. So let's get our toolkit ready and figure out which is which.
So here's the puzzle we're facing. We've got two beakers with what looks like plain old water. I mean, they're perfectly clear, totally colorless. But one of them is an acid, and the other is a base, or an alkali. On the outside, they're identical, but chemically, they couldn't be more different. So how in the world do we unmask them? Well, we're going to need a few special tools.
Our very first tool is probably the most famous one in all of chemistry: good old litmus paper. Think of this as our go-to for a quick ID. It's not going to give us the whole story, but it'll give us our first major clue.
And the rules for this test are super simple. You take a piece of blue litmus paper, dip it in, and if it turns red, boom, you've found your acid. On the flip side, if you take red litmus paper, dip it in, and it turns blue, you've got the base. It's a straight-up yes or no answer. Acid or base. That's it.
But see, that's also its biggest limitation. Litmus paper's great, but it's not very descriptive. It's like a witness telling you the suspect was tall, but not giving you their actual height. We know one is an acid and one is a base, but we have no idea how strong they are. And for that, we're going to need to upgrade our tools.
So let's bring in our next piece of gear to build the case, the universal indicator. You can think of this as getting a much more detailed description of our suspects. It doesn't just yell acid or base. It actually starts to tell us how acidic or how basic something is.
And the secret to using a universal indicator is understanding the pH scale. Just think of it as a ruler for chemicals that goes from 0 all the way to 14. Right in the middle, at 7, that's neutral, like pure water. Anything below 7 is an acid, and anything above 7 is a base. And the further away from 7 you get, the stronger it is.
So how do you use it? It's actually pretty easy. You just dip the little paper strip into the liquid and you'll see it change color. Then you basically play a matching game. You take your strip and compare its new color to a reference chart, and that chart tells you the approximate pH. It's a huge improvement over just red or blue, right?
But, and this is a big but, it's still just an approximation. You know, matching colors by eye can be tricky. Is that a pH of 2 or is it closer to 3? For something like checking your pool water, close enough is probably fine. But if you're a scientist developing a new drug, that tiny difference could be everything. To really solve this mystery with 100% certainty, we need the final tool in our kit.
And here it is. We're bringing out the big guns now, the pH meter. This is the high-tech, digital beast of the modern lab. No more colors, no more charts, just a precise, undeniable number. This is how we close the case for good.
So why is this our final answer? Simple. It gives us an exact numerical measurement. We just stick the probe into our mystery liquid and we get a digital reading, usually to 1 or 2 decimal places. This is the difference between an educated guess and a cold, hard fact. No more guesswork.
Okay, let's test our first unknown liquid. We put the probe in, wait a second for the reading to stabilize, and the meter says 2.9. That is way down in the acidic range. So we've confirmed this is our acid, and it's a pretty strong one too. Case closed on suspect number 1.
And now for our second liquid. The probe goes in, and the reading starts climbing and climbing all the way to 11.6, way up on the basic side of the scale. This is, without a doubt, our base. The mystery is officially solved. We know exactly what they are and exactly how strong they are.
But hey, before we pack up our kit, every good detective has a couple of specialist tools for very specific jobs, right? Well, in chemistry, we have specialist indicators that give these incredibly dramatic and obvious results, but only under certain conditions. Let's check out a couple of the most famous ones. Okay, now this is just cool. Look at phenolphthalein. In an acid, it's totally colorless. But the instant you put it in a base, bam, it turns this amazing, vibrant pink. There is zero ambiguity there. And then you have methyl orange. It's red in an acid, but put it in a base and it turns yellow. These are just fantastic when you need a really clear visual aha moment. So, let's do a quick recap of our detective toolkit. We started with litmus paper, which is great for a simple yes or no answer. Then we moved up to the universal indicator to get an approximate pH value. For the final, definitive answer, we used the pH meter to get that precise digital reading. And of course, we've got our specialists like phenolphthalein for those really dramatic color changes. Every tool has its place.
And that kind of brings us to a final thought.
Each one of these tools gives us a trade-off, right? Between simplicity, speed, and precision. So if you were the detective with one last mystery to solve, which tool would you reach for? Would you go for the quick and dirty litmus test, or would you trust only the precise, definitive pH meter? The right tool, it seems, always depends on the mystery you're trying to solve.